Hello, I'm Laura Cassidy from the American Chemical Society. Welcome to this media briefing from ACS Fall 2021. We're joined today by Dr. Magnus Wolf-Watz from Umeå University. He's using evolutionary time travel to reveal an enzyme's origins and possible future designs. Dr. Wolf-Watz?
Thank you, Laura, and also thank you for having me here on this media briefing. I would just like to start by saying that we know that most of the things that happens in our bodies are operated by proteins. So proteins are small, tiny machines that takes care of everything, all the way from degradation of food products to vision and smell. And as it happens, when we look to the complex life forms like humans or other eukaryotic organisms, we see that we have different organelles. And on those different organelles, we need different types of chemical reactions to occur. And chemical reactions are usually catalyzed by enzymes, otherwise life would go too slow. So we've been interested in how the fuel molecule that is called ATP, or a very similar molecule called GTP, is recognized by enzymes. And Laura, can you please show this first slide?
Sure, there we go.
Yes, so what we have here is, to the left, we have a cartoon of the most common fuel molecule that we have in our body, and that is ATP, that is used to drive all processes that require energy. And that molecule can be selectively recognized by an enzyme that we are interested in, something called an NMP kinase. And ATP is recognized with one single hydrogen bond that is formed, or one single interaction that is formed from the enzyme to the substrate ATP. And ATP is built up of a base, a ribose, and 3 phosphate atoms, as shown to the lower left of the panel. Now, in mitochondria, we have another version of the fuel molecule called GTP, and that is used in cell respiration. Basically, it is used to produce massive amounts of ATP, so one can also consider GTP as a common fuel molecule. GTP also has other specific roles in cells, like in protein synthesis or in eukaryotic organisms, also as cell signaling. And one thing that we've been interested in is in the evolution of this specific recognition of ATP versus GTP. It's just one difference in the recognition of a bond, a hydrogen bond donor or hydrogen bond acceptor. And in order to look into that, we got interested in the concept of time travel, because what we're doing is that we're looking back in time by understanding that there exists a evolutionary ancestor to eukaryotic cells. And presently, what is believed to be the latest ancestor to eukaryotic cells is a phylum called the Asgard phylum. It's an archaeal phylum. And within that phylum, there exist 4 different lineages. These are Heimdall, Thor, Odin, and Loki. And for several reasons, Odin archaea is believed to be the oldest evolutionary lineage. And to that end, we looked into a NMP kinase from this organism to try to understand how ATP and GTP selectivity could evolve in for instance, in human cells to take care of the functions that should be catalyzed in the cytoplasm versus the mitochondria. And Laura, can you please change slide? So what we did was that we isolated a NMP kinase from Odin archaeota. And we found that the enzyme, in fact, is a trimer. And Laura, you can start the movie now. So it's a 3 protein or enzyme molecules that are held together with a very distinct trimerization motif. So we are rotating the molecule here, and the structure is determined with X-ray crystallography. And if we zoom in here, we see that the recognition that is made to the substrate is actually done by the side chain of a glutamine. And that glutamine, as you can see here in orange to the upper left, actually can recognize any kind of NTP. So we have discovered a universal NTP binding motif. And this universal NTP binding has later diverged into the ATP and GTP binding that we see in the human cytoplasm or in the human mitochondrial matrix. So we think that we have found really the evolution, or one aspect of evolution that makes us the organisms that we are. And with that said, that is the main core of the discovery that I'm presenting on my talk on the ACS Fall meeting.
Thank you, Dr. Wolf-Watz.
Thanks.
And now I'd like to ask you a few questions about your work.
Mm-hmm.
Can you tell us a little bit more about this unusual form of life, archaea? And I understand it kind of has features of both prokaryotes and eukaryotes.
Yes. Basically, what we have is that we have three domains of life. We have bacteria, we have archaea, and we have then eukaryotic organisms. And it's been a pretty big debate on the evolution of eukaryotic organisms. What is the most or the oldest common ancestor for eukaryotic organisms? And it was this big discovery around 2015 by a group in Uppsala, where they found in a deep sea hydrothermal vent in the Atlantic Ocean a completely new phylum. And by doing analysis of this phylum that is called the Asgard archaea, it was discovered that it contained a lot of proteins that usually only are found in eukaryotic organisms. So these are called eukaryotic signature proteins, or ESPs. So that is one of the indications that is used to propose that they are the most or the closest ancestor to eukaryotic cells. And as you're right, as you're pointing out, that archaea contains features of both eukaryotic and prokaryotic organisms. For instance, it's been suggested that some archaea might contain filaments that usually only are found in eukaryotic organisms. So that is the discovery that made us interested in pursuing the structure determination and functional analysis of an enzyme from this specific archaea that is called Odin archaeota. Turns out to be a good choice.
Interesting. And are these archaea found exclusively in these deep sea thermal vents, or are they other places around the world?
There are, and one thing that is interesting with this deep sea vents is that they're quite often hypothermophilic vents or high temperature vents. So for instance, the enzyme that we studied had a melting point of close to a boiling point of water. So it was around 91 degrees. But there also exist other archaea that are living in more, let's say, moderate conditions like around 30 or 50 degrees. And they are then called mesophilic organisms. There exists actually a large divergence in the habitat temperatures for archaea. We can also find psychrophilic archaea that lives in colder temperatures. Our particular case was a hypothermophilic variant.
Why did you choose to study this particular enzyme, adenylate kinase? Is it an enzyme you'd worked with in the past?
We are studying it because we are basically what we are interested in is the following. In our cells, they're going on thousands and thousands of chemical reactions. If you take one of those chemical reactions outside of the cell, put it in a test tube, you will find that it is very, very slow. Many reactions are so slow that they will take thousands of years or millions of years. And of course, that cannot be. In life, things must go much faster. So chemistry must at least be much faster than cell division. That in, for instance, E. coli is around 20 minutes. So every chemical reaction must at least be much faster than 20 minutes, or must be executed within 20 minutes. And the solution, elegant solution to that, I would say, in organisms is to use enzymes that speeds up the chemical reactions. So enzymes are used to sort of merge the timescales of biology and chemistry such that they commensurate. And that is where my interest comes in. We are interested in the basic properties or the basic fundamental principles that makes enzymes so extremely efficient in speeding up the rate of chemical reactions. A lot of things are known, but still there exists a lot of things that are unknown. And I think that we were lucky or fortunate to find this universal multi-substrate binding enzyme. And that adds, of course, to the general knowledge that we have around enzymes.
Mm-hmm. You mentioned this universal NTP binding motif that you discovered in the archaea enzyme. Is this unusual, or are there other examples of enzymes that can bind all NTPs?
It's very unusual, and there exist other. There are some other, at least examples of enzymes that has broad substrate specificity. What we've been able to do is to go one step further, and that is to determine the structures at high resolutions of all these complexes. So we know the precise geometry of all atoms involved in this universal NTP binding motif. So it's long answer to that question. There exists a few examples, but I think that we have the most well-defined example, as far as I know.
Was it difficult or challenging to collect and purify enzymes from archaea to determine their structure?
It turned out to be a pretty straightforward operation. I had a postdoc that was working with this, and we had a huge advantage that the protein was hypothermophilic. So we can actually do our first, we produce most of our proteins in E. coli. And we could then just subject our E. coli lysate to high temperature, get rid and denaturate most proteins, and then we have our trimeric protein left. And then we were also lucky in a sense that it was quite easy to crystallize the protein or the enzyme, also with different substrates. We were able to crystallize the protein with 4 different substrates, and I think that was really nice for us. And here it was a multidisciplinary approach, actually working together with another lab that is doing crystallography with bachelor students, postdocs. It's been a pretty big project.
Okay. Why do you think the human enzyme evolved to no longer form a trimer?
Yes, that we have a very good answer to that. And it turns out that human enzymes, the human NMP kinase, are surprisingly active. They have an extremely high enzymatic activity. And that's been puzzling to us. But then we realized by comparing the structure of this trimeric archaea, we understood why. And what has happened during the course of evolution is that the eukaryotic enzymes has become monomeric. And by becoming monomeric, they can add one additional amino acid residue to the catalytic site or to the active site to get this boost in activity. The archaeal enzymes have to sacrifice that residue in order to make the trimerization motif. So by doing the trimerization, you need to sacrifice some of the components of the active site, and thereby activity goes down. So I think that we have a pretty good explanation for that. Is quite fascinating result that we got out from our structure determinations.
And I think you said the trimer might be more stable in the extreme temperatures of these deep sea vents. Is that also true?
It is. That is actually true. So we think that part of the reasons that the enzyme needs to be a trimer is to maintain a very high enzymatic activity. No, a very high melting temperature, I would say.
Okay.
The activity is low, the melting temperature is high.
How do you plan to use what you've learned by looking into the past to engineer novel enzymes? This is kind of the future part of the time travel.
Exactly. So that is exactly what we are working with right now. So what we think is that on a more general sort of perspective, protein design is a very important field of research that can be used to manipulate enzymes, to create enzymes that can catalyze novel chemical reactions or can be used in synthesis of complex drug molecules and such. And to this end, we need a lot of different building blocks that we can use in design. And we think that our general NTP binding motif can be such a generic building block that can be used. And what we're doing is taking our first baby step is by taking this universal motif and transplanting it into a human monomeric variant that is, for instance, ATP or GTP selective, and to try to make it multi-selective. So that's our first thing to do to establish that as a piece of building block that we can use. But then, of course, moving forward, there's a lot of things we can do with design. One thing that we're working with in our lab right now is degradation of the cocoon of the silk moth in order to produce silk fibers. That is also an enzymatic reaction where design could help to make that process much more efficient.
Interesting. What other enzymes from Odin archaea do you plan to study? And do many of their enzymes have human homologs?
We haven't really decided that yet. So what we're thinking here is that we came across what we call a treasure chest. We studied one enzyme and found this fantastic result. So we think also by studying more enzymes, we can learn much more about evolution and also other functionalities in in the protein universe. So we're right now, as we speak, trying to curate and make lists of what enzymes that we're going to address, but we haven't come to the final conclusion yet, and still work in progress. But it's really fascinating. We're quite motivated to do this work as well.
Sure. Okay, to conclude, what do you want viewers to take away from your talk? What's the key conclusion or message?
I think that a key message is that what we would like to do is to learn about features that makes us the organisms that we are. And one way to do that is to look into the past. It's like in history, right? If you're, I mentioned before, if you come from Rome, if you want to understand who you are, it could be a good idea to understand the Roman Empire. That's clearly affecting the citizens of Rome today as well. And the same goes with proteins. To understand proteins of today, modern proteins in eukaryotes, it can be a good idea to look into the past, to do a time travel, to try to understand what evolutionary aspects made enzymes the way they're operating today.
Very good. Thank you. Media briefings for ACS Fall 2021 will be posted throughout the meeting at www.acs.org/ACS Fall 2021 briefings.